Um, okay, it's a pity that Matthew cannot be here to give a seminar in person, but we're glad to have him over Zoom. Matthew is professor of statistics and human genetics at University of Chicago. And I think for the last 15 years, I've been an admirer of Matthew's work. His work has offered really deep insights in both human genetics and statistics and also computing. He has made his work sort of, has a flavor of building these models that are, beautiful models from the standpoint of statistics, but they're also very useful and practical in yielding real biological insights in the large genetic data sets that we are seeing. So, back then, a decade ago, Matthew's work has, his method structure has great impact in how we look at population admixture in genetics models. And recently he's just look, he's so many different works, but I'm just talking about the ones that I'm familiar with. He has a recent paper that gave new insights on noise models in single cell sequencing. And my lab has been working with his topics models and grade of membership models in looking at spatial and single cell sequencing data. So lots of different contributions in different parts of statistics and statistics, genetics, and genomics. So so I'll hand you the floor, Matthew.
Thank you so much, Nancy. Thanks for your kind introduction and the invitation to speak. It's great to be here. It's great to see some of you on Zoom. Hi, Ed. And tell you about this recent work. So let me share my screen. Um, so yeah, so this is joint work with several people in my lab, including particularly Gao, Peter, Abhishek, and Yu Chin, who are listed here. And so I wanted to start with kind of telling the story a little bit behind this work, what motivated us to do this work. So this here, we were involved in the GTEx project, the genotype tissue expression project, which essentially was a large NIH-funded project that collected gene expression data on lots of tissues in lots of individuals, and also genotype data, so DNA variation data on the same individuals. And one of the things they wanted to do was look for which genetic variants appear to be involved in regulation and specifically associated with or ideally causally affecting gene expression in the different tissues. So all the tissues have the same DNA essentially, but they express different genes and in different ways. And so part of the idea is that we might be able to understand that regulation process by identifying the genetic variants that are associated with gene expression in the different tissues. So this is a kind of a picture from the GTEx portal, or the data shown on the GTEx portal. It's a subset of the data. You don't have to be able to read the labels, but the different rows here are different tissues, and the different columns are different genetic variants, SNPs. And each SNP, these bars, the intensity of the color and the height of the bar tells you something about how strong was the association between genetic variants at that SNP with gene expression, a particular gene in that tissue. So that's what each. And so people use this kind of database when they've done a genetic association study, maybe in some disease they're interested in, and they find a genetic variant that's associated with their disease, and they're wondering, well, does this genetic variant do something? Can I say anything about what it might be doing? And one way they might try to answer that question is to go to the GTEx database, if you like, and say, well, is this genetic variant associated with gene expression in some tissue or other? And maybe we can get some insights that way. And what you're supposed to get from this picture is that there's a lot of red and to some extent blue as well. So blue is just where a particular, it just depends on the coding of the alleles, where the genetic variant is decreasing expression associated with a decreased expression instead of increased. But really the size is what matters here. And what you can see is there's lots of red, okay, which means there's lots of genetic variants that are associated with expression in lots of tissues. And I should be clear that this association is just marginal. It's just looking at one SNP at a time, one tissue at a time, and asking is it associated and how strongly is it associated. And of course, part of the reason there are so many associations is not because there are so many causal effects, but because there's a lot of correlations between these different measurements. So in particular, genetic variants that are nearby one another in the genome, like these ones are often very highly correlated with one another. It's important to appreciate the very highly correlated. What I mean by very highly correlated is correlation of 1, so completely correlated in the sample, or 0.995, very highly correlated. So highly correlated in the sample that it's going to be very hard to tell the difference between them. And they essentially give, you can see here, these two SNPs have the same results essentially, or almost indistinguishable, certainly visually indistinguishable results across all the tissues. And that's because they're, as a variable, they're almost identical to one another in the data set. On the other hand, if you look really closely, there are differences. Some of these bars are slightly lower than others. Right? So although they're significantly associated, one possibility is that, there's one genetic variant here that's actually driving all these signals. On the other hand, maybe that's not the case. Maybe there are multiple genetic variants in this region that are underlying this big sea of red. So our goal is to narrow down this kind of, these broad omnipresent associations, many of which are due to correlations between the different SNPs, narrow it down and try to say, well, what's a more parsimonious potential explanation for the data? Could we explain all this data by a single genetic variant that has an effect in all tissues or not? What kinds of, what do we need to explain these data to get it down to a narrower oh, sorry, I'm not advancing. Okay, yeah, so this is what I'm going to call fine mapping, this problem of narrowing down, these association signals to a small, ideally small, possibly small, the smallest we can reasonably do based on our statistical, what's supported by the data, set of potential causal SNPs that may drive these associations. And ideally, I want to do that by analyzing jointly across all the tissues simultaneously. But when we started thinking about how to do that, we realized that so there were already existing methods to do that in one tissue, but extending those existing methods to deal with, say, 50 tissues, which is what we've got here. Well, as I started to write a grant explaining how we were going to do that, I couldn't convince myself it was going to work because these methods are already pretty complicated and pretty computationally intensive. So what I realized was that we probably needed to simplify the process for a single tissue before we were going to be able to expand it and deal with 50 tissues. So actually today's work, as I said, there were already some methods that you could argue kind of give a reasonable solution to the problem for one tissue, although I'll also argue that we've made some progress there. But one of the goals was just to simplify the process enough that we would be able to expand it up to 50. And that's what we're in the process of doing now. I won't really tell you about the 50 version. I will just tell you about the single tissue version, which I think is already pretty interesting. But just to keep it in mind that one of our goals was to simplify this process so that we could complicate it again down the line. Okay, so so I'm focusing on a single tissue here. There's still lots of associations. Many of these variables are highly correlated with one another. So what's the explanation for this association? So the next few slides are just mostly to kind of introduce some basic frameworks and notation. So excuse me if it's fairly basic. But just to tell you that these association tests I've just shown you are usually done SNP by SNP. That's looking one variable at each time, and they fit a regression model. Y here is going to be the phenotype, that's the gene expression data, a particular gene, and XJ is going to be the genotype, the SNP, the genetic information at SNPJ, and beta j here is the coefficient of SNP-j in this simple regression model. So this is a simple regression model with a single SNP in it. Easy error term, we're going to assume that's normal. And the null hypothesis is that beta j is 0. And of course here beta j measures the association, the marginal association of SNP-j with y. So when there are kind of some really simple formulae for getting the beta hat and its standard error, these should look somewhat familiar to you. And we also may make use of a z-score. People often will compute a z-score and a p-value for each SNP, and that's kind of what that picture was I was showing, is basically the results of these z-scores for each SNP. Okay, something that may be slightly less familiar to some of you, but is still pretty straightforward, is that, and these are going to play a role later, is that you can compute a Bayes factor for each SNP. You have to make one additional assumption, which is a prior distribution for beta j under this model. So we assume it's a normal with mean 0 and some variance that I'll talk about a little bit more later. And if you do that, then you can get a closed form for the Bayes factor that measures essentially the ratio of the probability of the observed data under the hypothesis that SNP-j is associated divided by the probability of the data under the null that it's unassociated. So this is a simple function of that z-score and the standard errors and the prior. Okay, so this is gonna play a role later. The main thing to notice is it's closed form. It's simple. It's no harder to compute than a regular p-value, essentially. Okay, so that's what people do when they're measuring association, but when we want to try to get at causality here and find map, the technique we use is to look at all the SNPs jointly. So that's here we've done the multiple regression version of the model where all the SNPs are now in this matrix X. Many of the columns of X are highly correlated with one another, but the model is that we add up over SNPs their effects. And now I've used bj instead of beta j now to indicate that its interpretation is different. The interpretation of bj is that it's the association, or I'm going to call it the effect, of SNP-j on y after controlling for the effects of all the other SNPs. And if we make a few more assumptions that there are no unmeasured confounders, everything's typed, there's no population structure, then we can interpret bj as actually a kind of a causal effect of SNP-j on y, and that's what I'm going to do with these caveats in place. I don't think they're too burdensome. Actually, probably the most burdensome thing is that we don't always type all the genetic variants, so we have to keep that in mind. But we're going to assume that we do and kind of gloss over that detail, because even if we do that, the problem's still not quite straightforward. Okay, so we're going to be fitting this multiple regression model. And ideally, we're going to assume that B is sparse, and we're going to try to identify which elements of B are non-zero. So we framed, essentially, fine mapping, for those of you who didn't know the term, is variable selection in multiple regression. We're asking the question, which of these elements of B are non-zero? And maybe I just want to emphasize that in many statistical applications, we try to select variables because it can improve predictive performance. But that's not why we're trying to select variables. Here we really are trying to learn about a state of nature. Here we're trying to understand which genetic variants are actually influencing gene expression. And so that's the reason we're selecting variables. And I think it, in some sense it matters, because, in the prediction case, it doesn't matter on whether you select a variable with a very small effect or don't select it, that the effect on prediction will be completely the same, essentially. But in our case, we're really trying to interpret these selections as causal. So it's a little bit more. It's a different context, I guess. It's important to bear that in mind. Okay. So why is this a difficult problem? Well, let me just give you a cartoon example to explain what can happen with genetic data. So, as I've mentioned, in genetic data, it's not unusual for two of the columns of X to be extremely highly correlated. In the extreme case, of course, two columns are identical with one another. So in my example here, I'm going to assume that the first two columns of X are identical to one another, completely correlated. and that the next two columns of x are also completely correlated. X3 and X4 are completely correlated, but uncorrelated with X1 and X2, let's say. And let's say X5, etc., the rest are uncorrelated. So that maybe there are 1,000 columns of X, and they're uncorrelated with X1 to X4. And furthermore, I'm going to assume that there are two real effects here. The first one has an effect with size 5, and the third one has an effect with size 1. So, the idea here is X1 has a big effect, X3 has a smaller effect. And so, of course, because they're completely correlated with finite data here, we can't guarantee we're going to select the correct variable. We can't tell the difference between 1 or 2. So although I'm telling you 1 has the effect, from the data available, the best we can tell you is it's either 1 or 2, but we can't tell which because they're completely correlated in our data. And the same is true for 3 and 4. 3 and 4 are completely correlated, so all we can tell here is that you need 1 or 2, and you need 3 or 4, but I can't tell you whether it's 1 or 2 that you need because they're completely correlated, and I can't tell you whether it's 3 or 4. Okay, so the idea is I want a method that, given this kind of situation and given enough data on Y and a small enough or large enough sample, let's say, would come up with this conclusion. So this is what I mean when I'm going to talk about uncertainty in variable selection. When there is uncertainty, I want to extract that and communicate that. And another way of thinking about it, or phrasing the communication, is in terms of sets of variables. So I'm going to define what I'm going to call a 95%, or you can choose a different level, but a 95% credible set of variables as a set of variables that has a 95% probability to contain at least one variable that has a non-zero effect. So that's my definition. And here we're going to interpret probability in a Bayesian sense. So our goal is to report as many credible sets as the data support, each one as small as possible, and ideally non-overlapping. Although we won't require that strictly, but that's ideally. So in my example, we would report two credible sets: one that contains variables 1 and 2, representing our confidence that one of these two variables has an effect, but we don't know which one, and a separate set containing variables 3 and 4, representing our confidence that one of these has an effect, but we don't know which one. So that's another way of thinking about it, is we want to report. You can think of these as, if you think of it in terms of a kind of discovery, multiple testing type discovery type framework. These sets are our discoveries, and they're a true discovery as long as they contain a real a SNP with a real effect. But we also want them to be as small as possible. That's the additional kind of component to this. So it doesn't seem that complicated, I think, to a statistician when you put it like this. But on the other hand, actually existing methods don't really address this problem satisfactorily, or didn't when we did this work, I think. So you may be familiar with penalized regression methods, which essentially just select variables and give you a set of selected variables. And they do, in my example, they would just choose one of the first two and choose one of the second and third and fourth, and just give you a set. And they don't, even if they chose a different set, they don't, maybe they chose ElasticNet might possibly choose all four, but they don't give you any uncertainty about saying, oh, you need 1 or 2, but I don't know which, and you need 3 or 4, and I don't know which. It hasn't got the grammar to communicate that in the inferential results. And Bayesian regression methods, which can be done via all subsets or MCMC or stochastic search, they kind of do have the potential to communicate this kind of thing, but it's actually not straightforward to get from the results of a typical Bayesian regression analysis to the kinds of succinct summary that I just gave you, and I'll say a bit more about that in a moment. I'll just mention another thing that people do, which is to start off by just clustering very similar variables together and then test the clusters. So here are a couple of papers that have that flavor, and that's, I think, getting closer to what we want. But the problem with those is knowing how to cluster the variables in advance, and often these variables don't really form nice distinct clusters? And if two variables are at 80% correlation, do you put them in the same cluster or different clusters? And so there's a lot of kind of ambiguity about this first step. So essentially we want to avoid that first step of clustering variables, and simply the clusters kind of fall out in the sets at the end as we need one of these variables, but I can't tell you which one. So let me just say a little bit more about how the Bayesian approach to regression would proceed in this kind of toy example. So I've got my 4 variables here. Remember, 1 and 3 have an effect, but 2 is correlated with 1 and 4 is correlated with 3, identical to 3, and so we can't tell them apart. But if we used a suitable sparse prior, and let's assume we can do the computation well enough and we've got big enough data, as a cartoon, roughly speaking, what the Bayesian method would conclude is that it's one of these 4 models. In other words, it would have a model where there was at least 1 or 2 in there and at least 3 or 4 in there. And in fact, all of these models would have equal support from the data because they're actually essentially identical models, because 1 and 3 is identical to 1 and 4, is identical to 2 and 3, is identical to 2 and 4, because 1 and 2 are identical and 3 and 4 are identical. And of course it could include more, but the sparsity, I'm just kind of waving my hands a little bit and saying if the prior is sparse, we're going to favor these minimal configurations. And in principle, this information, the posterior probability on these 4 models being 25% each, actually says exactly what I want it to say. It says you need either 1 or 2 and you need either 3 or 4. If you stare at this matrix long enough, you'll convince yourself that essentially it's saying exactly that, because every model has either 1 or 2 in it and every model has either 3 or 4 in it. So in principle, this contains all the information I need and want, but actually going from this kind of list of models and their probabilities is already, even with 4 models in this very simple case, a conceptual step to say I need either 1 or 2, I can't tell you which, and I need either 3 or 4, I can't tell you which. And in practice, what we actually get from a Bayesian method, when you apply it to a typical fine mapping problem, is a list of 1,000 different models, each of which has very small probability. And you're left with the challenge of trying to extract from that list of 1,000 different models this kind of information about, well, which variables are actually causal and which ones can we not distinguish between. So in fact, what people do instead in a Bayesian context is to use these marginal summaries. So they use these posterior inclusion probabilities, which is just the marginal probability for each variable that it's included in the model. That is the probability that Bj is non-zero. So in this case, we add up the models for the probabilities on the models for which SNP1 is included, and we get 50%. We do the same for SNP2, same for SNP3, and same for SNP4. And of course, you get a 50% inclusion probability for every SNP, which is kind of right, but because it's a marginal probability, it's lost this information that you need either 1 or 2 and you need either 3 or 4, right? It's just because it's marginal. So although this is actually a useful summary and a good first start, we want to keep a bit more of that joint information around and give these sets. So that's why marginal summaries don't answer the problem. Let me also mention that the computations are often time consuming to run, also time consuming to code. And as I've kind of mentioned here, going from this output to simple inferences is hard, and these posterior inclusion probabilities are insufficient. So that's what we're trying to achieve, and that's why existing methods were not really up to the job for our purposes. So before I go on, maybe I'll just pause a moment and see if there are any questions about that.
Matthew, so in your definition of the credible sets, is there some kind of a set size requirement that the set needs to be small? Because if you make the sets big enough, they will always be 95% credible.
Yeah. So we don't make a requirement in the size in our definition. We just define it like that and then say, ideally, we compare methods on how small the sets they produce are. And if two methods both produce sets that have a good coverage, that is, have this property, they often contain 95% of the time, say, contain a causal variable, the one that produces smaller sets would be the better method. So there's no requirement in the definition, but we will use that criteria as we would ideally like it to be small. And in fact, sometimes our method does output very, very big credible sets that contain essentially all variables. And we typically just throw them away because we say, well, those may or may not be right, but they're not much use to us anyway, whether they're right or not. So, there's I'll mention that later, but it's a feature of at least some forms of our method that it sometimes produces very, very large sets containing almost every variable, and we just discard those. Thanks for the question. Okay, so to build up to how we're going to solve this problem, let me just mention, or let me describe this rather simple solution, which is to assume that exactly one variable has an effect. So it's kind of a ridiculously simple assumption. Actually, it turns out not to be that ridiculous in some genetic association studies, because the effects are very sparse, and maybe in some regions it's not that implausible to assume that exactly one SNP has an effect, but we would certainly like to relax that assumption later, and that's really what our work's going to be about. But for now, let's just notice that if we make this assumption, all our problems disappear, and let me explain why. So we're going to fit this multiple regression model, but we're going to assume that B has exactly one single effect. So I'm going to call that exactly one non-zero effect. So I'm going to call B a single effect vector. Some people call it a one sparse vector. So it has one non-zero element. So the only question is, which one is it? Which element of B is non-zero? And we can compute, using our Bayesian framework, we can compute a Bayes factor for each SNP and the posterior inclusion probability for each SNP, as I kind of outlined. These are all analytic calculations, very fast. And essentially, this PIP gives you the posterior probability that each variable is the causal variable, and because there is only one causal variable, that tells you everything you need to know. And furthermore, a credible set is easy to compute because you just line up these pips from the biggest one to the smallest one, and you form you add SNPs to the set until the cumulative pip is 100%, or 95% rather, 95%. So in this case, everything's straightforward. So in my toy example, let's just see that in my toy example. In my toy example, there were two effects. So I'm going to simplify it. I'm going to just assume that there was only one effect and see what happens. So let's suppose only SNP1 had an effect. The pips would be 0.5, 0.5 on X1 and X2 because they have, they're completely correlated. X1 has a strong effect, so X2 is also strongly marginally associated. They're completely correlated. We can't tell which one it is. So they both get pip 0.5, and we correctly get a credible set containing both variables. So in this toy example with one effect, the single effect regression, that's the regression it's a multiple regression, but with only one non-zero effect, gives you everything you need. And the reason it's so trivial to fit is that under the assumption there's only one non-zero effect, there are only p possibilities where p is the number of SNPs, so we just enumerate them all. We can just do exhaustive enumeration, and it's very fast. It's linear in the number of SNPs. And of course, what we'd like is to generalize this single effect regression to allow more than one effect. And the usual approach is to simply allow more than one of the BJs to be non-zero, but that immediately creates all these computational challenges of now we've got J choose L models to enumerate, where L is the number of non-zero effects we're going to allow. And so our new approach is essentially, it's a trick. You can think of it as a reparameterization, simple reparameterization of the model that's going to make it easier to do these calculations. So, okay, so let me write down the model. So instead of having, we've still got y equals xb, but instead of allowing b to directly have more than one non-zero effect, we're going to write b as the sum of L vectors, each of which has one non-zero element. So each BL here is a single effect vector. It's a vector with one non-zero effect. We don't know which one it is, has the non-zero, but the assumption is that BL has one non-zero effect. And so, and by summing capital L of them, let's say capital L is 10, if we want to allow for up to 10 non-zero effects, this vector, this sum of vectors will have up to 10 non-zero elements. If you could allow for 100. Typically, we're looking at problems where there's a small number of non-zero effects, so 10 or 20 might be typical in our applications. Actually, smaller than that might be typical, but we often allow up to 10 or 20. And think of L as an upper bound on the number that we expect to find. Okay, so it's just a reparameterization. So why is it useful? Well, the reason it's kind of useful is it suggests an nice iterative fitting scheme. The idea is that if you knew all the BLs except one of them, sorry, this should be a capital L here, there's a typo. If you knew B1 up to BL minus 1, then fitting the last one is now just a single effect regression problem. You have one non-zero effect, you don't know where it is, and so you can use the single effect regression procedure I just outlined. And then, well, you don't know B1 up to BL minus 1, so it suggests essentially a simple iterative procedure that go through fitting each of the BLs in turn, essentially assuming the others are known.
Is there an order problem? Do you have to pick the order here?
So no, because they're all the same, so it doesn't matter which one. I mean, they all start at 0. So there's a natural starting point, which is that everything's 0. And essentially what you're going to see is it's going to look a bit like a forwards regression, stepwise regression. I'll tell you a bit more about that.
Thank you.
So this is the analogy, right? So you might be familiar with this very simple, probably much denigrated fitting scheme. So I'm not sure how. I'm going to argue that our method looks a bit like this, but fixes some of the problems with stepwise selection. So stepwise selection essentially fits, for each SNP fits the marginal, the simple regression, just fit Y on that SNP, and it selects the best variable among all the variables. So it just does the marginal test, uses something like a p-value or some measure of fit to choose which one to include, and then it forms the residuals and repeats until some criteria is met, right? So you do all the marginal tests, you choose the strongest one, you regress that out, and you repeat until there's some kind of stopping. So this is a greedy algorithm, so that's kind of somewhat problematic. And also at each stage here, it's just choosing the best fit and then repeating. So what our algorithm is going to look like is like we're calling it Bayesian stepwise selection, or actually we call it iterative Bayesian stepwise selection. So the Bayesian stepwise selection would, for each SNP, fit this model by computing a Bayes factor. And then instead of choosing the best fit, we basically form a weight for every SNP that's proportional to the Bayes factor. Those were our pips we did before. And we select, instead of selecting the best one, we kind of think of selecting each SNP with probability proportional to its weight or its pip. We don't really randomize that selection, I should say. They're kind of, just think of the selection event is a fuzzy event that assigns a weight to each SNP. So it's like a partial selection of each SNP. And then we regress out the weighted version of the XB and repeat until bored. But we can also go back to earlier and refit the model, so you can make that can make up for bad you might pick the wrong thing, or the wrong thing might have a big weight in the first step, and you can come back and down weight it. We'll see an example of that later. So let's just see how this would work in our toy example. We're familiar with this. What happens with forward selection? Well, in forward selection, the first step, because x1 has a big effect, it would select either x1 or x2. It wouldn't know which one, so it would just select one, and then it would move to the next step, and at the second step it would select either x3 or x4, and it would just give you a pair, and that was it. The Bayesian version, at the first step, it would select each of x1 and x2 or assign them weights 50-50. So it would be kind of a fuzzy selection event and say, it would tell you that I don't know which one to select. It's x1 or x2, but I don't know which, and I'll give them 50-50 probability. And then at the second step, it would then regress those out, and that would be essentially the same as regressing any one of them out because they're completely correlated. And then at the second step, it would kind of assign 50-50 probability to each of 3 and 4, and it would say, I don't know which of these to select, and it would keep that information around. So it would report at the end one credible set containing x1 and x2 with 50% weight each, and a second credible set with x3 and x4 with 50% each. Okay, so that's the idea in a nutshell. So you might have at least two questions now. One is, does it actually work in practice? And the other question you might have is, what's this algorithm actually doing? Can you kind of give me a bit more of a justification than this heuristic or analogy with forward selection? So I'm going to try and answer those questions in turn. So firstly, I'm going to pick out a kind of challenging example from our simulations. This simulated data where there are two, and show you how it works on this. So we'd simulated data where there are two causal SNPs, so the red ones and the black one, the red ones, SNP1 and SNP2 here. They both have an effect. There are 1,000 SNPs. These are real genotype data, so they show these complex patterns of correlation. And I picked this simulation out because the marginally most associated SNPs, that's these, so the y-axis here is just the marginal log 10 p-value for each variable. So the marginally most associated SNPs are not the causal SNPs. And the reason this happens is that these variables are correlated with both SNP1 and SNP2 in a way that kind of boosts their association, so they show a stronger marginal association than either SNP1 or SNP2 because they're associated with both. And I should say this, I've cherry-picked this simulation to be this difficult example. Most examples actually, one of the highest marginal associations will actually be a causal SNP. So this isn't necessarily the typical setting. It's picked to illustrate a problem that we feared might happen. And although there's no guarantee, most of the time, and in this particular example, the algorithm works fine. Let me show you what happens. So this is the two sets it identifies. The first set identifies two SNPs, one with most of the weight, which is the actual causal SNP, but it's only got, say, 90% of the weight. So to get to 95, we have to include this in the credible set. And then it includes these variables here, which are all kind of up here, very strongly correlated with one another. It can't tell you which of these is the causal SNP, but it's confident that one of them is, and indeed, one of them is. So that's the output. And so why does it work? Well, what happens is that in the first iteration, so here I've got the variables along the x-axis, and I'm showing the alpha is like the weights that I said. And this is after the first iteration. We're setting L equals 3 in this example, and so at the first thing it chooses is this red one, and these are those marginally strongest associations. Of course, by definition, it's using the marginal associations to make its first decision. So these red ones show you the weights of the SNPs in its first iteration of its first pick. And then of its first effect, so that's L equals 1. Then it tries to pick a second SNP, and when it does that, it picks up this signal, because even after it's found these associations, there's still something remaining to be explained. And then once it's picked these, there's still something remaining to be explained. So it picks one of these, and one of these is actually the SNP1 in the previous plot. And then we iterate. So when we iterate, after 3 iterations, what happens is now it's picked out these green and blue SNPs. It realizes these red ones don't look as significant anymore, because essentially it starts to control for these green and blue ones. So in the iterative procedure, the red ones start to get down weighted, and by iteration 8, the red weights are really down. The green ones are, it's decided this one's probably the causal SNP for the blue ones. It's still uncertain about the green ones. And by iteration 18, what it's done is what Nancy was saying before. It's actually spread the red weights out among a very large number of SNPs, because of course we know there are only 2 effects here. And once it's found them, the green one and the blue one, it doesn't know where to put that last one anymore, because actually there is no effect. And so what happens is, if you run this algorithm on null data where there's no effect, all the sets tend to be very big, which is kind of a symptom that it doesn't know where to put the effect, and so you can kind of just throw them away. Actually, so maybe I'll just pause there before I show you simulation results, which I might largely gloss over, but I'll see if there are any more questions about that.
I was just a little confused when we saw all those red ones. Were you now, was it putting equal weights on all the red ones in the very first iteration?
So in the first iteration, right, it's the weights are, it's selected about what, 10 or 15 SNPs here with high weight. I mean, there are 1,000 SNPs, right? And it's selected about 10 or 15 with highish weight. They're not all the same weight. Their base factors are all slightly different from one another.
Uh-huh.
Yeah. So
And how did it know to do 15?
Oh, well, it just computed these weights proportional to the base factors. So
I see.
Right. So it's got mass 1 that it spreads. I mean, formally it's spreading it across all the SNPs, right?
Ah,
But
okay.
most of them get negligible weight.
Got it. Thank you.
So there is red down here, but it's so close to 0 you can't see.
So are those 15? Those 15 are the highest base factor? Is that?
Yes, exactly. They'd be a subset of these guys, right? So if you just looked at this, it's going to give the most. I mean, it's using, although I've put p-value here, the base factor and the p-value are very closely highly correlated with one another. So these guys are also going to have the highest base factor, and so they're going to get the highest weight in that first selection event. Yes.
And so the trick is,
okay, and this is, I mean,
you're just doing 1,000 individual regressions. Yeah.
Yes, exactly. It's doing 1,000 individual regressions, computing a base factor for each.
Great.
And then and weighting them. Yeah.
Great.
And then it regresses, then it removes the effect, quotes the effect, the estimated effects of those SNPs, and repeats that process. At which point these are the ones that get the weight.
Got it.
And you can see, yeah, and then it repeats that process again after removing those, and these are the ones that get the weight. And you could just stop here, and in that case, this would be the wrong answer because of this greedy problem, right?
Right.
It would be misled by that initial false step, and it doesn't always happen. It can get stuck in bad solutions like this, but it can escape them as well.
Matthew, I think I'm a bit lost about what you do in the second iteration. So in the second iteration, you have the 3 different
right.
colors.
Okay, so we've picked these. We've got the 3. We've got each selection event which has weights on each SNP. So we chose one. We weighted every SNP and we regressed out the effect. Essentially, that's the same as a weighted version of. It forms the weighted residuals after regressing out each. Actually, all of the 1,000 SNPs, it actually regresses out all 1,000, but then weights them by their weight. So most get negligible weight to form the residuals. It does that again to get 2. It does that again to get 3. So then at that point, assuming we're going to stop at 3, we could go to 10, but let's just stop at 3. And now we're going to go back to the first one. We're going to add back in the effects that we took out, so that we've only removed 2 and 3 now, these, and we're going to re-estimate B1. So
I see. Okay.
in this model, we're kind of regressing out B2 and B3 and re-estimating B1. Yeah. And this process does converge, which is like maybe I'll get to that after I show some quick simulations. I guess that's just one example, but let me show you some simulation results to, I don't know. So these are 3 different MCMC based or stochastic search based methods for doing the same problem. So these are more conventional, let's say, approaches. Each of them has a slightly different computational approach and a slightly different prior, but they're all kind of trying to fit the same model essentially. Okay, so their posterior inclusion probabilities ought to kind of agree with them, and they ought to kind of agree with ours. So this is just looking at the posterior, all the methods output posterior inclusion probabilities. So I'm using them to just do this comparison here, not because I think that's the most important thing, but all the methods output those. So they're a way of comparing methods. And we're doing simulations with 1 effect variable, 2 effect variables, or 3 to 5 effect variables. And you can see when there's only a single effect variables, all the methods are really agreeing pretty well. And maybe that's not surprising, it's a very simple problem. This method, DAPG, is slightly got some slightly odd behavior. Probably is a stochastic search method, so it's probably not searched hard enough or something, which is leading to these elevated pips. For some, why I should say the red ones here, you can't really see the color very well, but these are all black. That means they were not actually causal variables in our simulation, right? And then this is with 2 effect variables, and you can see that actually most of the pips are right down here near 0. So this scatter actually kind of overemphasizes the difference between the methods. Actually, the methods mostly are agreeing. Most of the variables are here, and there's a lot along the line, but there's some, disagreement. And when the methods disagree, one thing to note is that there's more red points below the line usually, and that's our method. That's where our method is assigning a larger pip. So when the two methods are disagreeing, generally speaking, the larger pip is being assigned to a causal variable by our method. So these are examples where we assigned a pip of 1, but it kind of missed it. You can think of the others as almost gold standard. I mean, as I say, this one's doing exhaustive enumeration. So for what it's worth, of all models with less than 5 variables or something. So in some sense, you might think of that as a gold standard. It takes a very long time. This is ROC curve for these things, and that's our method at the top. So actually, I don't think the ROC curves are that different, but for what it's worth, ours is competitive. This is the coverage, empirical coverage of the credible sets. So this is a 95% nominal coverage, and the red one is our method. So you can see that if there's one causal variable, we've got coverage over 95%. If we simulate 10 causal variables, the coverage is around 92% in these simulations. So it's there or thereabouts. Obviously, I'd rather it was 95%, but there is no guarantee because it does depend on the prior. This is DAPG, which is another method that kind of came out at a similar time to ours and produces something similar, not quite exactly, that you can kind of call a credible set. You can see its coverage is slightly lower, but not too bad. Really nicely, it's very fast. So this is per data set. It's taking about half a second. The other methods take quite a bit longer. This is the exhaustive enumeration, which is why it's so slow. And the computation scale linearly with the number of samples and the number of variables and the number of non-zero effects that you include. Okay, so this is really the last thing I want to tell you, which is what is this algorithm actually doing? So what it's actually doing is computing or optimizing a variational approximation to the posterior under our model. So here are our variables. These each are single effect variables. So this is a vector of length p, remember, which has one non-zero element. So these are the variables in our model, the parameters in our model, I should say. And what variational approximation does is to assume that the posterior has some simplified form, and in common is to assume that they factorize somehow. So what we're really doing under the hood here is assuming that the posterior over the b's factorizes in this way. But it's really important to say this is not factorizing over variables. This is factorizing over L. So if capital L is 10, it's factorizing into 10 components here. Each one of which, each QL here, is a distribution on a vector of length p that has exactly one non-zero element. So the distribution QL is basically saying what's for each element, it has a probability of it being the non-zero element, and it has a distribution on how big it is, conditional on it being the non-zero element. So this is a distribution, it's a p-dimensional distribution, but it's a p-dimensional distribution on elements that have only one non-zero element. And that is the key, is it doesn't factorize over variables. We and others in the past have fit variational approximations to linear regressions where we did exactly this, but we factorized, instead of over L, we factorized over the p variables. And that has exactly the wrong properties for this particular variable selection problem, because it's saying that if you've got two variables that are very highly correlated, and you don't know which, the right posterior is I don't know which one. We only need one of them, but I don't know which. And that's not independent, right? That's a highly dependent distribution. It's of the form, we need one of them, but we can't tell you which is exactly what this QL is capturing. So it's this reparameterization and then use of if you're familiar with variational approximations, then you probably could have skipped everything I've said about the intuition and just say, oh, I can just reparameterize the model and do this variational approximation. And then if you do a gradient ascent, or coordinate ascent rather, on the QLs, you get exactly the algorithm I described. So essentially, the algorithm I described is optimizing QL, the variational posterior approximation on BL, keeping each of the others fixed. Okay, so if you didn't understand that, let me say that what it tells you is that the algorithm has an objective function. Every iteration of the algorithm is guaranteed to increase that objective function, and so it will converge, but potentially to a local optima of that objective function. Okay, so it could, so going back to Ed's question about order, we don't really think of it as order being a problem, but you could imagine that if we started from a different point than we do, we would get a different answer. Actually, so since the slides and the paper were produced, we've come across a few more situations where it didn't work as well as we'd like, and we were a bit more motivated to try and find something a little bit better. And so we actually have some kind of refinement procedures to try to diagnose convergence to local optima and escape them when they happen. But it works most of the time actually out of the box, but that refinement can help a bit in complex situations. Okay. The elephant in the room maybe is, how do I deal with how big L is? And let me just say two things about that. Firstly, I've kind of already said one of them, which is if you set L to be 10, but there are only 3 effects, what happens is that it creates a large number of credible sets where it just doesn't know where to put the effect. And so it basically spreads them out across, it spreads the effect across hundreds of the 1,000 variables in our simulations. And so one strategy is just to say, well, those credible sets may contain a real effect, but they contain almost all variables, so they're not much use to us, and just throw them away. Another slightly more principled approach, which we also have implemented and use as kind of our default, is to do an empirical Bayes estimation of the prior variance for each effect. So essentially, to put a prior for each effect, I'll have a variance parameter that's the prior variance, and to estimate this as part of the inference scheme. And what happens is then, if you set L too big, some of these get set to 0. The estimates are 0. So it prefers to set the effect to 0 than to spread it out among all of the variables. Doesn't always happen, but it's this device filters out most of those big, avoids most of those big ones automatically, and then we still filter out the large ones automatically. Before wrapping up, let me just tell you what happens for this one example. So this was the example I gave you at the start. I took the data from this tissue here. As you see, it's red all over the place. And the answer is the data look like it's consistent with just a single causal effect. And there are 5 variables in the credible set. So we can narrow it. Essentially, we can narrow down all this red and these highly significant marginal p-values to 5 we think it's one of 5 causal SNPs. And we don't think you need any more explanation. Of course, we're not saying there aren't any more effects. We're just saying there are none that we can confidently find. Oh, you can use this not just for fine mapping. So this is change point detection. Maybe I'm running out of time, so I'll just say that the effects here correspond to the change points in this location, and the red here are our credible sets for the change points. So each credible set contains one of the true change points here, and this, for example, this change point, you're a bit more uncertain about where it occurred than this change point because the change point is smaller, so it's harder to see and it's harder to be sure exactly where it occurred, so you can do that. And the paper maybe contains an example of, I mentioned it can converge to local optima, and this is an example where the black line is the fit but the red is the truth. And the reason it converges, it converges to a fit with no change points. And the reason it does that is you really need 2 change points in this example to explain the data. One isn't enough. So iteratively fitting it by adding 1 change point and then doing it one at a time just doesn't work very well. So this is kind of I made this example to be especially challenging, and indeed it doesn't work for this example. So I'm just going to go straight off to say we've got software and a paper, and open the floor for questions because I'd be really happy to have any.
Could I ask?
No. Oh, go ahead, Ed.
Oh, good. So I just have one. So this is absolutely spectacular stuff. But I was wondering, what about situations where sometimes, for example, each variable by itself won't work, but they're highly correlated, but together they're both needed?
Yes, I guess that's exactly here what's going on here, right? That you need both change points to fit the data. If you just put one of them in, it would actually be a horrible fit to the data, right? Because so what this is a change point where everything's going on, and then there's a short change point that goes up. The mean goes up for a couple of steps, and then it goes down back to the original level. And if you can't fit this data with 1 change point, you'd do worse than 0 change points. But if you put both in, you can actually get a better fit. And the truth was there are 2. So this is exactly the kind of setting where, okay, so I should say the variational approximation is still capable of capturing this, and the criteria we're optimizing is optimized at the right solution. But the simple forward, we call it iterative Bayesian stepwise selection, our simple algorithm for optimizing doesn't work.
Right.
It gets stuck in a local optima.
So yes,
your example is one where our algorithm would not converge very well. Although, the criteria is like an MCMC, right? MCMC is, if you run it, it might also struggle to find those settings, right? Because most MCMC schemes work by changing one thing. And that's exactly the kind of setting where they can have mixing problems, but if you run them long enough, they'll eventually find it. Okay, we don't have a algorithm that will eventually find it, but we do have a criteria that if you optimized, it would find it. But
thanks.
Yeah, I see a question in the chat from CDB. The question is, I wonder if Matthew has an estimate for what fraction of association signals yield such a synthetic association caused by partiality between 2 causal variants.
Oh yeah, 100%. That's easy. Yes, so LD in almost every variable has at least one other variable that's very highly correlated with it by some definition. Now, however, you could ask a different question, which is of what proportion of the time can we narrow things down to a single causal variant, right? How often can we, some of our credible sets will contain a single variable, which suggests that we've narrowed it down to a single one. I can't remember that fraction. Sorry, but the paper will give you an exact. The larger the sample size, the the better the narrowing down you get for a given effect size. Anyway, essentially, what matters is the significance. So a high marginal z-score will give you the highest ability to narrow things down. And there's some non-trivial fraction of associations that get narrowed down to a single variable, but I can't remember. Maybe it's 20%, maybe it's 50%. I can't really remember. It will depend on the GWAS, the sample size, how significant things are. But it's, yeah. Did that answer your question, CDB? See, you still have your hand up.
I think, is there any other questions? I think we're at the end of the hour. Is there one more question? No? Okay. Well, thank you so much, Matthew, for a great talk. And
Yeah, thank you for having me.